Thank you, Catherine. And before I begin, yes, thank you to the organizers for inviting me. I hope everyone can hear me okay. Good. Let's see if I can get this started. And can you see the presentation? Yes, okay, thanks again. So yes, I'll be talking a little bit today about migration, diet, and health in the colonial Caribbean, and specifically with a focus on new insights from isotope bioarchaeology. So a little bit about the underlying theory or the theoretical approach. I'm taking what we call a life history approach to construct individual osteobiographies. Sometimes these are referred to as isobiographies if we also incorporate isotope data. And in keeping with the sort of theme of the lecture series on material culture, I'm drawing on the work of Johanna Sofauer on the body as material culture. And in some cases, as I hope to show later, this is sort of very literal, that there's certain cultural practices that leave a clear imprint on our bodies, things like cranial and dental modification. In other words, in other cases, it's perhaps less literal, but more in the sense that a lot of our activities, a lot of our life ways and life experiences are imprinted on our skeletons, and sometimes in ways that are visible macroscopically, but in other ways that are invisible unless you use very specific biochemical or isotopic techniques. In general, life history approaches, and osteobiographies in particular, tend to draw on information from archaeology, when available, also the documentary records, so historical and oral historical records. And the corpus of what we rely on is generally osteological analysis, and in more recent years, other types of biomolecular methods, things such as ancient DNA and isotopes, which is the primary focus of today's talk. So I'll give a little bit of background about the underlying principles of isotope methods and the methods that I used. I'm assuming that everyone sort of heard a little bit about isotopes, being archaeologists, but I'll just briefly mention them so we're all on the same page. So the main underlying principle of isotope analysis in bioarchaeology anyway is that you are what you eat isotopically. And what we mean by this is that amongst various isotope systems, these vary in the isotope ratios that they have in the environment. So anything from soils to water to the atmosphere to the foods that we eat. And these are ultimately reflected in the isotope values of human tissues or animal tissues for that matter, via the mechanism of diet, primarily for the things that we consume on a daily basis. Sometimes these isotope values are identical to the environment. Sometimes there's a little bit of a shift, but we can calculate what that is because we know the underlying principles of how they work. But ultimately, the idea is still the same. So if you analyze the isotope values in human tissues, you can say something about not only the diet, but also other aspects of the environment where that person lived. It's also important to keep in mind when we're talking specifically about human tissues is that different tissues reflect different time periods of our lives. So in general, things like our teeth, particularly the enamel of our dental crowns, but also the dentine of our tooth roots, these generally form very early on in our lives, starting with our baby teeth as early as in utero, when we're still in our mother's womb. And our teeth tend to mineralize and form throughout childhood. And once they're fully formed, they don't alter that much, at least from a chemical point of view. So teeth can fall out, they can break, they can crack, they can wear down. But chemically speaking, they're very robust and they don't really change after they form. And so in that sense, they're really ideal for these types of approaches because they sort of lock in the chemical and isotopic signature of the place where they were formed. This differs very distinctly from, for example, bones. The different bones of our bodies. Bone is a sort of living tissue, unlike dental enamel, which is rather inert. And because bone is a sort of composite matrix of organic and inorganic components, it's a living tissue, and so it will adjust over time to new living conditions. And the exact way the rate that it adjusts or equilibrates to the new environment changes or varies from person to person and also from bone to bone. And so in that regards, bone reflects more of a sort of long-term average over years or even decades of life, but more towards the end of life. There are other tissues that grow at a very regular rate. They tend to grow very incrementally, things like hair and nails. And these can be important sources of information for like the end of life, the last several months or years of life. Unfortunately, these are hardly ever recovered in archaeology, but they can be sometimes in cases, for example, in permafrost, mummies, bald bodies, and so on. I won't go into any more detail about that, This entire presentation focuses primarily on the skeletal system, but I thought it was important to note because these have important implications, for example, for forensic isotopic approaches. Okay, so what is then isotope bioarchaeology? In this regard, I'm talking about bioarchaeology more in the North American sense, so the study of human remains from archaeological contexts, and isotopes referring to all the different isotope systems or measurements that we can make of these human skeletal remains. Now, these methods are actually quite old now, starting as late as the late 1970s. And they generally divided into two broad categories. In general, strontium and oxygen are used, at least when they're applied to human skeletal remains, to investigate things concerning human mobility and migrations. So things like migration patterns, distinguishing locals from non-locals, and in some cases, determining an individual's origins. Strontium isotopes, very simply, generally reflect the underlying geology of where someone comes from. I'll get into a bit more details about that in a minute. Whereas oxygen primarily reflects climatic and geographic conditions. In contrast, isotopes of carbon and nitrogen, they're very widely used in archaeological science approaches, particularly to the bioarchaeological record for many years now, and these generally reflect different aspects of food waste. So in particular, carbon isotopes can allow us to distinguish between the consumption of terrestrial versus marine foods, and in certain cases, from the consumption of C3 versus C4 plant foods. I'll talk a little bit more about that in a minute. I guess the most important thing that we need to know about C3 and C4 plant foods is that the vast majority of plants taxonomically are C3 plants, and they tend to have low carbon isotope values, whereas there's a very small number of economically important plants that have use a different photosynthetic pathway, and they have much higher carbon isotope values. But I'll explain that also in more detail in a second. And lastly, nitrogen isotopes says something about the trophic level of a specific organism. When applied to human remains, I guess you could interpret this more as the degree of carnivory or the distinguishing between plant consumption versus meat consumption. Now, in general, for most of the last several decades of isotope bioarchaeology, these first two proxies, strontium and oxygen, have been treated very differently or separately from carbon and nitrogen isotope approaches to the study of diet. And only in fairly recent times, I'd say the last decade or perhaps less, that people started to integrate these different isotope systems more holistically to look at the sort of complex interdynamics between migration and diet, which is sort of the main thrust of this presentation. Now, for those of you that are not familiar with it, just a quick refresher, how does strontium work? The strontium, at least in geological deposits throughout the world, it displays a lot of variation, primarily based on the underlying lithology of the bedrock, but also the age of the bedrock. Strontium enters soils through the weathering of minerals, and from there it's uptaken via groundwater into plants, and thereby strontium enters the food chain. Those plants are ultimately consumed by animals and by humans. And strontium is very, very similar to calcium, so it's an alkaline earth metal. It's non-toxic at normal concentrations. Has a similar ionic radius to calcium. So anywhere where you find lots of calcium in your body, you will find measurable amounts of strontium. So most of the strontium in our bodies is concentrated in our skeletal system. In other words, our teeth and bones. Now, if we consider a sort of simplistic ecosystem where nobody is consuming foods that were imported long distances, which is not always the case, as obvious from the previous presentation. But if we assume a sort of a simplistic ecosystem, in theory, then every living organism within that ecosystem should have a similar or even identical strontium isotope value. And so what that means is that if you grow up in a place, you'll have a strontium isotope value characteristic of that location. And when you move, eventually your bones and your hair and your nails and the rest of your body will ultimately equilibrate over many years to the strontium isotope value of the new location, but your teeth will always stay the same isotopically in terms of their strontium isotope. And that's why it's sort of a very good, what we call a passport of people's place of origin. Oxygen is quite different from strontium. The main underlying principle has to do more with climate and geography than underlying geology. Oxygen is one of the most abundant elements on Earth, and most of the oxygen in our skeletal system ultimately derives from the consumption of water, either directly through drinking water or the water contained in the foods that we eat. The main source of water vapor in the atmosphere comes from evaporation occurring on large bodies of water, particularly in the tropical or warm places on Earth. And by definition, those have the highest oxygen isotope values. And ultimately, what happens is through iterative processes of evaporation, transpiration, and precipitation, the oxygen isotope values of water vapor tends to decrease as the water vapor moves across the Earth's surface or throughout the atmosphere, moving away from the water bodies, moving away from the ocean to northern, high latitude or low latitude locations, or as water vapor moves inland from the coast to more inland locations or to more high altitude locations. So this also ultimately results in very specific distributions of oxygen isotope variation across the globe. Once again, consuming water throughout our lives, the oxygen isotope values of our teeth will sort of lock in the climate, the environmental conditions of the place where we grew up. And if we move, once again, our other tissues will ultimately equilibrate to the new location. But once again, our teeth will lock in these values. And then lastly, carbon and nitrogen isotope systems, which are probably the more commonly known ones. These really reflect different patterns of diet and nutrition. And they primarily work, as I mentioned before, based on a few very simple principles. So nitrogen primarily increases as we move up trophic levels within an ecosystem from plants to herbivores, omnivores, and carnivores. We call this process trophic level enrichment. And there's a sort of typical offset or increase in nitrogen isotope values when we move up these trophic levels within the ecosystem. Carbon isotopes, by contrast, they tend to reflect two main different sources. In terrestrial systems, they can reflect differences between C3 and C4 plants, shown here on the bottom. So C3 plants tend to have very low carbon isotope values, and C4 plants tend to have very high carbon isotope values with no overlap between them. And as I mentioned before, C3 plants, it's a sort of primordial or primitive situation. So at some point in time, all plants on Earth were C3, and then eventually C4 plants evolved, and they were very much adapted to sort of semi-arid warm climates. They're still quite rare overall, and there's only a very small handful of economically important C4 plants that are consumed by humans. And these include maize, also known as corn, very important throughout much of the Americas in prehistoric times. Millet, which is an important staple crop in many parts of North Africa and Northeast Asia. Sorghum, also common in parts of Northern and East Africa, and of course, sugarcane. So depending on what environment or ecological system we're looking at, carbon isotope values can be used to distinguish, for example, between the consumption of C3 and C4 plants. And in other cases, they may reflect differences in the consumption of terrestrial versus marine foods. And as humans tend to be sort of hyper generalists, we tend to eat anything. How you interpret the carbon isotope data depends a lot about, or reflects a lot about the ecological system that we're looking at. So you have to sort of really have a good understanding of the surrounding ecosystem in order to interpret human isotope data in this regard. Okay, now moving on to the case study. This is a site on the north coast of eastern Cuba. It's known as El Charro de Maíta. It's a fairly large cemetery, at least by Caribbean standards, so over 110 individuals. The vast majority of the individuals that are buried here are considered to be of indigenous peoples. These are known more commonly as Taino, although most Caribbean archaeologists don't use this term, but in the common discussions of people around the islands, they're referred to as the Taino people. These are more or less the people that inhabited most of the Greater Antillean Islands when Christopher Columbus so-called discovered the Americas. The site itself was believed to be occupied for a fairly short amount of time, roughly between 1450 and 1550 AD, so somewhat contemporaneous with the previous site in Sweden that we just heard about. And the site was believed to be part of the encomienda system, and this was a sort of the first system of labor exploitation imposed by the Spanish conquistadors of the Caribbean. And it was a sort of not entirely enslaved labor, but a way of controlling the indigenous populations that had been conquered. So Columbus arrives in 1492. Shortly thereafter, he starts setting up the first European settlements on Hispaniola. Quickly thereafter, they conquer some of the other surrounding islands, including Cuba in 1510. And right around that time, they start to impose this encomienda system. What that basically means is that in exchange for Christianization of the local populations and their so-called protection, they're obliged to give a certain number of months of labor to the Spanish conquistadors and settlers, primarily to work in mining camps at the beginning and later on much more focused on agricultural production. The most important thing about this particular site are two main things. The presence of a cemetery itself, as you can see in the bottom right here, is actually somewhat odd. There were no indigenous cemeteries in other parts of the Caribbean at this time. So the cemetery itself is a sort of Spanish or European introduction. Most indigenous communities in the Caribbean primarily buried people either in caves or underneath their house floors. So the fact that it's already a cemetery says something very special about this particular location. And secondly, it's one of the rare indigenous populations that we know that spans this very last period, or this transitional period from the late prehistoric to the arrival of the Europeans and the conquest and colonization of the Caribbean. So in that regard, it's very, very unique. It allows us to say something about this really dynamic period of colonization and migration that was going on in the world at this time. Now I'll go try to walk us through the isotope results one by one in the simplest way as possible. So we thought this is a really ideal population to apply this sort of multi-isotope approach to understand how things like mobility and diet, health status, colonization, and so forth all sort of are interlinked or potentially interlinked with each other. So this first chart simply shows the strontium isotope values on the y-axis, and the x-axis, the results are just aligned from lowest to highest. So there's no important information on the x-axis. It's just for visualization purposes. And we've also measured a number of local animals to try to get an idea of what the local range is. And that's generally how we interpret strontium isotope data. We want to characterize what we think a human should have, a local human should have for that particular area. And we characterize or estimate the local range, and then people that fall way outside of that range are considered to be potential non-locals. And for this population, roughly about 25% out of the 88 people that we measured were considered non-locals, 22 in total. Most of them had isotope values that fell below the local range, and there was a handful of people that clustered slightly above the local range. And there was one person here with a really, really high value, upper right here. And what's particularly interesting about this individual is that we know that the strontium isotope values for the islands of the Caribbean are fairly well characterized based on previous studies. And basically, there's no location in the Caribbean at all that has strontium isotope values higher than about 7093, which is the value of modern seawater. So basically, when we find individuals that have isotope values above that line, roughly, we know that person is not only not from the site, non-local to the site level, but also non-local to the island, and actually non-local to the entire Caribbean archipelago. They must have been, by definition, a long-distance migrant. In general, we believe that the vast majority of people that are buried here are indigenous people, indigenous Taino people. The non-locals are probably also indigenous Taino people, but most likely from other parts of Cuba or other islands nearby, because they sort of more or less have isotope values that are consistent with the Caribbean in the broadest sense. This fairly high rates of migration, 25%, also is consistent with what we know about the European colonial system. One of the first things they actually did when they conquered Cuba is they started moving people around from place to place. So people generally were quite distributed into small villages. They brought them together into much larger concentrations. And they also started moving people, for example, from the Bahamas to other places because they needed essentially labor to do mining, to do diving for pearls and so forth. Now I'm going to zoom in on two particular non-locals, the one I mentioned before with the really high isotope values, and one of the individuals with one of the lowest strontium isotope values. Beginning with the individual with one of the lowest strontium isotope values, individual 72B. In addition to this low strontium isotope value, this individual also had a very low oxygen isotope value in their dental enamel. Very high carbon isotope values in both their dental enamel and in their dentine and bone collagen, which I'll show in a minute. It was an adult female with a generally large stature, so she was over 10 centimeters taller than the average for adult females at the site. And she displayed very unique patterns of dental and cranial modification, which you can see here in this picture here. So cranial modification was quite widespread in the Caribbean, although not universal. But this particular mode of cranial modification had never been observed before in the islands of the Caribbean, whereas it is very common in certain parts of Mesoamerica. So that was already sort of a red flag that perhaps this person was not an indigenous Taino person, but someone that was perhaps a long-distance migrant. And the same also goes for the dental modification practices. Dental modification is entirely absent from the pre-colonial Caribbean, the prehistoric populations of the Caribbean. There's a small number of enslaved African burials that have dental modification, but once again, in a very different style than what we observe here. And once again, this dental modification style is quite common in parts of Mesoamerica, particularly in the areas around Belize and the eastern Yucatan. In addition, this individual displayed a very exotic burial treatment. She was buried face down with a very large boulder placed on her legs. This was a burial mode that we had never observed before, once again, for the Caribbean. And so the combination of all these different lines of evidence led us to propose this individual probably was a long-distance immigrant from the Maya region that had been transported, so forced migration from the mainland of Mesoamerica to the island of Cuba, primarily or most likely in the context of the indigenous slave trade. The second individual, burial 45, you can see here on the bottom right, had the highest strontium isotope value, and as I mentioned before, so high that it could not have come from the Caribbean. Once again, a very low oxygen isotope value and also very high carbon isotope values. An osteological assessment of the skeleton indicated that it had an African or sub-Saharan African cranial morphology. And what's interesting is that it was given a Christian burial treatment. So what's really interesting, another interesting aspect of this cemetery is that it shows a very interesting mix of indigenous burial practices where people tend to be buried in a fetal position with little or no grave goods, often on their side. But there's also a mix with a large number of indigenous people buried in with a sort of Christian burial treatment with an alignment east-west, arms crossed over the torso. And sometimes we also find these little metal pieces around their body. These are brass aglets, and these are essentially clothes fasteners that were very common in parts of Europe at this time. These are essentially analogous to a button, which was used to fasten clothes together. So several of them were found with this individuals, indicating that this individual was buried with European clothing. Probably based on all this filed evidence brought together, we proposed this individual was a West African, so an enslaved African migrant, once again brought over as part of the slave trade. Possibly one of the earliest enslaved Africans in the entire Caribbean. And based on this evidence here, we suspect had been Christianized in the process. Now, I'm going to zoom in a little bit on the isotope results, just to sort of explain how we came to these conclusions, and also to give indication how the isotopes for both diet and mobility can be combined together to tell a more in-depth and nuanced story. So the box here on the upper left indicates the measurements we have for indigenous populations from the Antilles. The isotope systems that we're looking at here is carbon on the x-axis and oxygen on the y-axis. Carbon, once again, says something about diet, and oxygen should say something about climate and geography. And we see here very clearly that this individual that we interpret as possibly Mayan falls way outside of the main cluster of individuals for the insular Caribbean, the islands of the Caribbean, as does the African individual. They both have oxygen isotope values that are lower, but not extremely different. But what's particularly interesting is their carbon isotope values are extremely high. And so we regard this, the most likely interpretation of this is that when these individuals were children, they were eating a large amount or high quantities of C4 plants. But the actual C4 plants are quite different. So that also says that you have to know something about the possible places of origin in order to interpret the dietary data. In regards to the Maya individual, we're almost certain that this comes from the consumption of high quantities of maize. Maize was the most important staple crop in Mesoamerica and most of the parts of Middle America and at this point in time. In fact, so much that the Maya actually worshipped a corn god. So it was literally a super important part of their lifeways. And in West Africa, we know in certain parts of West and Northern Africa, both millet and sorghum were extremely important staple crops. And so this African individual was probably consuming large quantities of millet and or sorghum as a child in Africa. Okay, now we're moving on to the carbon and nitrogen isotope data. The carbon is displayed here on the x-axis and the nitrogen on the y-axis. And once again, nitrogen indicates something about the trophic level. As we go higher with the nitrogen isotope values, that indicates increasing trophic level. And on the x-axis here, as we move from left to right, we see increasing consumption of either marine or seafloor plants. And plotted here is both bone collagen data, those are the red X's, and dentine collagen, which is the black plus symbols. And it's important to keep in mind something I mentioned earlier is that our dentine, which is the roots of our teeth, they form once again in childhood, but really don't change very much afterwards, whereas bone continuously remodels throughout our life, so it reflects a long-term average towards the end of our life. And what's really, there's two main things I want to sort of illustrate here or stress is that there's an enormous amount of diversity in this population, which is somewhat surprising given it's a small little village on the coast of northern Cuba. So when we look at, for example, the typical population in Northwest Europe, anytime from the beginning of the Neolithic to roughly the early medieval period, the amount of variation is about 2 per mil for both nitrogen and carbon. So that's about an entire population would fit into this little tiny circle here. Yeah. So it's a pretty small population with an enormous amount of dietary diversity. And this also sort of gives an indication that these people were being brought to the site from different parts of Cuba, from different parts of the Caribbean, and in this case also from much further afield, people from the Mesoamerica and also from West Africa. Now, if we look carefully here at the bottom right, we can see the isotope values of the two individuals that I'm talking about here. Once again, this person of supposed Maya origins here, there's their dentine and there's their bone value, and the individual of purported African origins with, once again, their dentine, their childhood diet here, and the adult diet here. Now, because the adult diet is so different from the rest of the population of El Chorro de América, and by a lot, it also tells something about how long they were at the site before they died. So if they had lived in Cuba or at this near or around the site for a very long period of time, for many years, we would expect these bone values, these red X's, to actually start drifting more and more towards roughly the cluster we see here for the local population. And that didn't happen. And because we also measured multiple teeth of these individuals, I've only plotted one here, but we know that even their third molars, which is the last teeth to form, also have non-local values. We can give a rough indication of when they migrated and how long they lived there before they died. So these are both adult individuals that died roughly between the ages of 25 and 35 years of age. And because their bones never really equilibrated to the local signal, whereas their strontium isotope values of their third molars is non-local, we can say that they must have migrated after the age of 18. And they could have only lived in Cuba probably several years before they died, which once again is in keeping with the very poor living and health conditions of enslaved labor in the Caribbean throughout much of the early colonial period. Now, one of the next logical questions that we ask once we identify these non-local migrants, of course, is where exactly are they from? We have all these, in this particular case, lots of lines of evidence indicating these non-local origins. But once again, it's difficult to say specifically about where they're from. So I've talked about West Africa. West Africa is enormous, larger than the entire Caribbean. Unfortunately, unlike many other parts of the world, for example, Europe, even parts of Mesoamerica, we don't really have a good understanding of strontium isotope variation in West Africa. And it's very difficult to assess that. It costs a lot of time and money. Fortunately for us, a researcher named Vicky Ulza, who's at the University of California, a few years ago received a very large grant to start to assess strontium isotope variation for Western Africa, and she's making very good progress. As you can see on this map here, these are all the locations she's already sampled. So I assume that we'll have a very good or much better understanding anyway strontium isotope variation in West Africa quite soon. And then I can maybe say with a much higher degree of certainty and precision where this particular individual may have come from. I also wanted to point out that the site in Cuba is not the only one where we've identified possible first generation forced immigrants from West Africa. There's a large number of colonial period sites in the Americas that have taken a similar or identical approach. And we often tend to see the same pattern. The local range is quite low, and we get an enormous diversity in strontium isotope values amongst these enslaved populations with really high strontium isotope values. That tells us two things. It tells us that any individual that we measure there with a strontium isotope value above the local range is most likely a first generation migrant. The fact that they're enormously variable is consistent with European strategies of enslavement of these populations. They were intentionally drawing people from different parts of Africa and from different tribes, often people that didn't speak the same language, and putting them in the same work conditions. They think that there was a conscious strategy to reduce the chances that they could have a slave revolt or rise up against the plantation owners. And also, we can ultimately then use these exact strontium isotope values once we have a better map of West Africa to say something about where these individuals come from, where they were actually born and raised before they were enslaved. I also wanted to mention something explicitly about these really high carbon isotope values for this particular individual from El Chorro, number 45. Because he has such a high carbon isotope value, we know that he must have been consuming a lot of C4 plants as a child. And we know a little bit about dietary variation in West Africa. And essentially, they have three main zones here. Here on the far coastal area, we have this rice zone, which is a C3 plant. Most of the other rest of sub-Saharan Africa, this is what we call the viticultural zone, where root crops and plantains were the primary staple crops. These are all C3 plants. So both of these areas would have really low carbon isotope values. And this area that's sort of highlighted in the shaded in the light gray is where C4 plants were staples, primarily millet. So once again, we see here that if we understand a little bit about the diet, this can also inform us about potential origins. And what I argue is that this individual must have originated from the Sahel zone of northern West Africa, this sort of semi-arid region where millet and sorghum are staple crops, and not from these more southerly locations where rice and root crops are staples. Okay, and lastly, moving on to trying to identify more specifically the origins of this Maya adult female that I mentioned before, individual CF72. One of the main ways we go about this is characterizing the spatial variation of bioavailable strontium and oxygen. And so we've made a lot of progress in this regard in the last few years, not only in the Caribbean, but other parts of the world as well. Essentially what we do is we build predictive models, maps of spatial isotope variation for the isotope proxy in question. And then we test these against the real world data. So we go out and sample lots of different types of animals and plants and so on, different samples to get a good idea of how the model works. And then we revise the model. And eventually we have some confidence that they reflect what the actual variation for that isotope system ought to be. Now in this image, we see the oxygen isotope variation for the circum-Caribbean displayed on top and the strontium isotope variation displayed on the bottom. Now, unfortunately, the oxygen doesn't display a lot of variation in the circum-Caribbean, which is sort of logical. It's all tropical to subtropical. It's all fairly close to the coast. And there are some mountains, but they're quite limited. What that means is that oxygen isotopes is not a very good proxy for identifying migration within the Caribbean. There's too much overlap, but it can be an interesting isotope proxy for identifying long distance migration from outside of the Caribbean. So things in the context of the slave trade, but for example, also identifying people from North America or from Europe that migrated to the Caribbean. Strontium is a different story. There is quite a bit of strontium isotope variation in the Caribbean, although limited perhaps on a global scale. Now, what we wanted to do is see if we could combine these two maps, what we call isoscapes, so maps of isotopic or isotopic landscapes, and see if they could tell us more specifically or more precisely about the origins of these particular individuals. I'm focusing here specifically on this individual CM72B, where I've already argued that we think that she was coming from the Maya region. What you see displayed here in the map is a very simple method that we applied. We simply took the strontium isotope value that we took from this person's tooth, And we eliminated all places on the map where the strontium isotopes did not match. So that the end result is all the places that are highlighted in pink, as you can see here, are places of possible origin based on strontium. And then we repeated the exact same process for oxygen. So we looked at all the places on the map where oxygen values were consistent with that that we measured in this person's tooth. And so individually, they're not very precise. Lots of different places are lit up. But when we combine them, that's what you see here in the purple, it increases the precision of the method quite a bit. So we're able to reduce or exclude many more different places just based on these two isotope measurements. And it's much more precise than one isotope measurement alone. And ultimately, then we can use the other evidence that I presented before in terms of cranial modification, dental modification, burial practices to narrow it down even further. And then I don't know if you can see it here in this sort of gray circle, green circle, excuse me, green square, I should say. This is where all the sort of evidence points to the same location. So all the isotopes values, the high carbon indicating high maize consumption, the strontium isotope values, the oxygen isotope values, cranial dental modification practices, and the mortuary treatment all are consistent with this particular part of the Maya region. So we argue that we have fairly good evidence now to suggest that this is the place where this person came from. And I should say that more recent but unpublished ancient DNA evidence also supports that this individual is an individual of Mayan ancestry. Okay, to wrap things up before we run out of time, I'm taking an isotopic approach to sort of construct these individual life history, these osteobiographies. And by doing a sort of careful selection of what isotope proxies you use and sampling different types of multiple tissues, and then taking on board lots of different types of evidence from archaeology, ecology, geography, we can start to answer some sort of fundamental questions about these individual life histories. For example, who migrated? Where did they migrate from? At what age did they migrate? What is the effect of the migration on their dietary patterns? And then ultimately, the next step I'm sort of focusing on is the influence of migration on their long-term health outcomes in terms of the age at death, pathologies, and so forth. I think that is where I wanted to wrap up. I wanted to thank you everyone for their time and all of my collaborators that I've worked with over the years on these projects. Thank you.